Hi everyone, my name is Piyush Kumar Pathak, and in this lecture, I am going to discuss one very important topic for NEET and other medical entrance examination, that is control and coordination. So, let's start with the video. Okay. So the name of the topic is control and coordination. The flow of the video will be in flowing order. First, we will have a brief introduction, what we are going to study. Brief introduction about the chapter. Then we will compare and contrast nervous system and endocrine system. Then we will look at the classification of classification of nervous system. Then we will read the structure of a neuron, structure of a neuron, and then we will look at the classification of neuron. So let's start with the introduction. So the name of the topic is control and coordination. So what does this control and coordination mean? To understand this, let's take an example of exercise. So when we exercise, what we are doing, put our muscles to we put our muscles to work. So when we are putting our muscles to work, we are using oxygen, because when muscles will use, they will require more oxygen. Also, they will require more nutrients. So nutrients consumption also increases. And when muscles will work, more waste product will be generated. Waste will be high, and also body temperature body temperature also increases. So to compensate the amount required oxygen demand, increase in required oxygen demand, what happens? Our breathing becomes breathing becomes short and deep. Also, when the nutrient requirement increases, what happens? Our heartbeat becomes fast. Sorry, flow of blood flow of blood increases when production increases. It will also blood flow to the kidney increases, so it is also similar, blood flow to the kidney. And when body temperature arises, we sweat. So to increase oxygen supply, what happens? This step is putting our lungs to work. This step is putting our heart to work. This step is putting our kidney to work, kidneys to work, and this is putting our skin glands to work. So this is putting our. So we are saying that. So we are saying that when we are putting one muscle, so when we are putting our muscles to work, it is also including heart. It is also including kidneys. It is also including lungs. It also including our skin and many other systems. So to coordinate the work of one muscle, many other organs are involved, and this is called coordination. This is called coordination. So if anyone asks us to define coordination, then we can define it as a process. It is a process which com- which complements and integrates different organs to perform a work. Okay, So coordination is a process in which com- in which organs complement and integrate each other to perform a work. Okay, so this is coordination. Okay. Okay, now let's move on to the next topic, that is, so if you ask me coordination, so for coordination there are two system in our body. One is nervous system, I will write it as NS, and we have endocrine system, and it will write as ES. So we have CS and NS and ES, endocrine system and nervous system. So what are the differences in their function? Why there are two systems? Because they are working differently. For example, nervous system is point to point connection, point to point connection, like bulbs, like switches in our home. We have different switch for different equipment. Similarly, there are different nerve points for different muscle groups or different organ. Similarly, but endocrine, it has a broad range effect. It effects can be in a large area of organ, or it is localized in a very local area, local part. Localized, it can be a broad range effect or a localized effect. The basic structure that is working in case of nervous system is neuron. So neuron are basically working in case of nervous system, but in case of endocrine system we have hormones. We have hormones. So hormones are responsible for functioning of endocrine system. The next difference is that it works in forms of electrical signal. It working in the form of electrical signal. So electrical signals are generated in case of nervous system, but in case of endocrine system we have chemical signal. We have chemical signal. Okay. Another difference is that the effect of nervous system is short lived. It is short lived. If we want to lift our hand, we will lift our hand and immediately it will go down. As soon as the signal is lost, the effect will be lost. But in case of endocrine system, the effect is long lived. It is long term effect. It is long term effect. And next point is that in case of nervous system, the things can be reversible. It can be reversible. If any effect has taken place, it can be reversed easily. It is reversible. But in case of endocrine system, it is mostly irreversible. Once the effect has changes have taken place, it cannot be reversed very easily. It is irreversible. It is irreversible. So these are the differences between nervous system and endocrine system. So first we will learn about nervous system, and in subsequent videos we will learn about endocrine system. Okay. So now let's move on to our next topic, that is nervous system. So in this video and subsequent videos we will focus on nervous system. Once nervous system is complete, we will focus on endocrine system. Okay. So nervous system. So first let's take a look about the evolution of nervous system. So in case of simple organisms like Hydra, it has the nervous system is very simple. It has nerve net. But when we go down the hierarchy, in case of cockroach, it has a brain, it has a brain, and ganglions, ganglions. And when we go down more down the hierarchy, we have mammals or humans. And in case of human, it has brain, it has a spinal cord, and it also has nerves. So it is very, so the brain nervous system of humans or mammals is very complex and advanced. It is very complex and advanced. But we are mainly focused on the nervous system of humans in this chapter. So let's take a look at the classification of nervous system of human. Okay. So nervous system of human or mammals is this. So the nervous system is broadly classified into central nervous system, and peripheral nervous system. So we have peripheral nervous system and central nervous system. We will write it as CNS and it will write it as PNS. So we have CNS and PNS. Okay. CNS consists of two organs, that is brain and a spinal cord. Brain and a spinal cord. And PNS consists of all the nerves that come out of nerves coming out of brain and spinal cord. So all the nerves that come out of brain and spinal cord consist the peripheral nervous system. We will learn about these nerves when we will study, when we will discuss spinal cord and brain. Okay. Till now we will looking at the classification. So peripheral nervous system is further classified into two categories. One is somatic, somatic nervous system, somatic nervous system, and another is autonomic, auto somatic and autonomic nervous system. So somatic nervous system controls our, it controls our, it controls our skeletal muscle, it controls our skeletal muscles, or we can say we can say voluntary muscles, voluntary muscles. So voluntary muscles or skeletal muscle are controlled by somatic nervous system, and autonomic nervous system controls our involuntary involuntary muscles or smooth muscles. Smooth muscles are controlled by autonomic nervous system. And this autonomic nervous system is further classified into sympathetic sympathetic nervous system and parasympathetic nervous system. So sympathetic nervous system controls emergency situations, emergency emergency. So sympathetic nervous system controls emergency body actions. So sympathetic nervous system controls emergency body actions, whereas parasympathetic nervous system controls our normal or baseline functions. So baseline functions are controlled, or you can say housekeeping functions. Housekeeping functions. So housekeeping functions or day-to-day function or baseline functions are controlled by parasympathetic, and emergency actions are controlled by sympathetic nervous system. Okay. So this was the whole classification of our nervous system. Now let's move to our Next topic, that is nervous tissue. Nervous tissue. So nervous tissue is classified. So nervous tissue consists of two things. One is neuron and another one are neuroglial cells. Neuroglial cells. So nervous tissue consists of neuron and neuroglial cells. So neuron are basically structural and so neurons are structural and functional unit. Functional unit of nervous system. Whereas neuroglial cells have many other functions like other than transmission of nerve impulse. They are basically involved in packing. They are involved in protection. They are involved in insulation, and they are involved in repairing. Okay. So some examples of neuroglial cells are astrocytes, and we also have oligodendrocyte. Oligodendrocyte. We have microglial cell, and we have swan cell. There are many cells. There are few I am naming here. So what is the function of astrocytes? They form blood brain barrier. We will be surprised to know that the blood cannot directly enter into the brain. So brain is not connected by blood vessels like other organs. There is a barrier here and blood diffuses through this barrier into the brain. So this barrier is found by astrocyte. Okay. And oligodendrocytes form myelin sheath. I will discuss one myelin sheath. Myelin sheath in CNS. Now you know CNS. Microglial cells act as phagocytic cell, and it helps in protection, like Kupffer cells in our liver. This swan cell also form myelin sheath in PNS, peripheral nervous system. The myelin sheath is formed by swan cell. So these were some neuroglial cells and their function. And now we will discuss our main hero, that is neuron. So let's take look at the structure of a neuron. So the structure of neuron basically consists of three parts. One is dendron, or plural is dendrite. Then we have axon, and then we have cytone. Okay. So let's try to draw the structure of neuron. So it will appear something like this. Okay, and we have these dendrites, Don't mind my drawing. Let's try to understand the concepts. Okay, so this is the nucleus, and we have some other things in this cytoplasm. I will tell about them. And this is the ground matrix, and we have the jacket. Okay, so this is basically a structure of neuron, and this is, let me change the color. So this is dendron, or dendrite is plural, and this central part is cyton, and this is nucleus. This is nucleus, and this black dot here, this is Nestle granules. Nestle granules. Then we have this as cytoplasm. Then this part, this part here, it is axon hillock. This is axon hillock. This is axon inside tube-like structure. This jacket is myelin sheath. Okay, and the myelin sheath is not continuous. There is gaps in between, and this gap is called node of node of Ranvier dot not Ranveer, and this is the axon terminal. This is telodendria, and this is the bulb synaptic bulb. Okay, so this is basically the structure of a neuron. These are their parts, and let's discuss about each part individually. So the first part is dendron. Let me change the color of the pen. So the first part is dendron. So dendron is basically involved in, first of all, these can be many, these can be many, and these are short processes. These are short processes. They are basically involved in uh, reception of, these are basically response, reception of signal or nerve impulse, and this also contains cytoplasm, and these have also neurofibril, and it also has Nest granules. So these are about the dendron. So Nest granules are basically rough endoplasmic reticulum. We have not learned about rough endoplasmic reticulum in cell chapter. So these are basically involved in protein synthesis. And as it is rough endoplasmic reticulum, it has ribosome on its surface. So why it was called Gneiss granules instead of rough endoplasmic reticulum? Because this was discovered by a scientist named as Nissl. So honor the scientist, it is called as Gneiss granules. And this is neurofibril. It is involved in centripetal conduction of impulse. So this neurofibril is involved in centripetal conduction of signal. Okay. So these are the about dendrons. Next is cytone. Cytone is also called as cell body or soma or perikaryon. Perikaryon. So there are many names for cell body. Basically, it is called cytone to match rhyme with dendron, cytone, and axon. So if we talk about cytone, this is the main region, central region. So this is the central region, and it has a nucleus, single. It has single. It has single. It has single nucleus. It has cytoplasm. It also has Nestle granules. It also has Nestle granules. It has neurofibrils, and it has and it also has but and another many point it is lacks centriole. Centriole is absent. Very important point. Centriole is absent, so neuron cannot divide. Neuron cannot divide. Therefore, we need to protect our head. If a neuron is damaged, it cannot divide again. The number of neurons are fixed. Number of neurons are fixed for the whole lifetime. Number of neurons is fixed. It cannot divide because they don't have centriole. Mature neuron don't have a centriole. That's why they cannot divide. They can only be repaired if repairable. Okay. So this is all about cytone. You know the function of neurofibril. It helps in the centripetal, or you can also write here internal conduction. Internal conduction. Okay. So this is the function of neurofibril. So we have central region, we have nucleus, single nucleus, we have cytoplasm, we have nucleus, we have neurofibrils, and centriole is absent. Please remember centriole is absent. Now we have axon. So axon is always single. Always remember it is single, and it is the longest of all the processes. So basically we can say that one dendron grew in length and it become axon. But its function is also different. It helps in transmission of transmission of signal. That is, it takes the signal away from the look dendron. It's uh, bringing the signal towards the cytone, and it is taken away from cytone. So its function is transmission, and its function is reception. Its function is reception. So there is a difference in function. So it is a single. It is always single. It is longest process. It helps in transmission of signal. It uh, it may and may not be myelinated. I will talk about myelinated. It can be myelinated. The the cytoplasm inside the axon is called axoplasm, and its membrane is called axonal membrane, exoplasm. And this exoplasm lacks neurofibril. Neurofibril is not present in axon. It is absent in axon. It has large number of mitochondria. It has large number of mitochondria. You will understand why it has large number of mitochondria. Okay. And the end of axon contains large number of vacuoles, at end, or you can say synaptic bulb. Okay, so this was all about the part of neuron. One point is myelination. So let's talk about myelination. So let's talk about myelination. So myelination, this is the myelin sheath. This is the myelin sheath. This whole thing is myelin sheath. So myelin sheath basically insulates the neuron. You will understand why we need insulation when we'll discuss nerve impulse conduction. Till now, so myelination is the basically process of formation of myelin sheath. So process of formation of myelin sheath is called myelination, and so myelination is done by oligodendrocyte in CNS. So if I ask you who does myelination in CNS, it is done by oligodendrocyte and Schwann cells in peripheral nervous system. Okay. So myelin sheath is basically a phospholipid, and we know Lipid are bad conductors. And if you ask me particular which phospholipid it is, then I will say it is sphingomyelin. It's very simple, sphingomyelin. So the name of the phospholipid is sphingomyelin, and it forms the myelin sheath. And this myelin sheath is formed in form of concentric circles. So this myelin sheath is formed in the concentric circle fashion. So layer by layer it is formed like this. So myelin sheath is formed in layers around the axon. Okay. And this myelination is not continuous. This myelination is discontinuous. So this myelination is discontinuous, and we have node of Ranvier where there is no myelin sheath. The point is called node of Ranvier, not Ranvier, it's Ranvier. Okay. And if a neuron is myelinated, it will appear as white. And if non-myelinated, and if it is non-myelinated, it will appear gray. So when many neurons group together and their axons are bundled together, they will appear white. And this group of neuron, we can say axon bundled, will form which one? Myelinated neuron. If they are grouped and bundled together, they will form white matter. They will form white matter. And non-myelinated are grouped together, they will form gray matter. So we'll learn about gray matter and white matter when we will study brain and spinal cord. Okay. So myelinated, this appears white because lipid is white. Sphingomyelin is white, so it appears white. And if it's not myelinated, there is no myelin sheath, it will appear gray. And if they are bundled together, it will form the gray matter. So now you understand white matter and gray matter. Okay. So this is all about the neurons, and now we will learn classification of classi, classification of neuron. So neurons are basically classified into two parts. One is on the basis of function, function, and second is on the basis of structure. So they are classified on two bases. One is based on function, another is based on structure. So on the basis of function, there are three types of neuron. First is sensory neuron. Second one is motor neuron, and third one is interneuron. They perform function of both sensory and motor. So what they are sensory neurons? They take signal from receptor to CNS. So they are taking the signal. So suppose this is a CNS. So this neuron is taking the signal. This is a receptor. This is receptor. So the neuron which is taking the signal from here to here is sensory neuron, and motor neuron takes the signal from CNS to receptor or effector muscle, whatever we say, effector muscle. Okay. So if this is CNS, then the signal is taken away from, sorry, it is taken this way. So this is sensory and this is motor. Okay. And in between there is exchange of information from here to here. This is done by interneuron. So this was the classification based on function. And now based on structure, number of processes, how many processes are present in the neuron. So based on number of processes, there are basically three types. One is unipolar, this, and second is bipolar, third is multipolar. So basically unipolar neurons have only one process. They have only one process, that is, and one process I already told you, that is only axon, because axon is only one. So that one process will be obviously axon. So this is example of a unipolar neuron. This is a diagram of a unipolar neuron, and it will be found in our embryo, because in basically they are formed in embryo. Then we have bipolar neuron. They have two process, one axon and one dendron. So this is the figure of a unipolar, bipolar neuron. So we have nucleus here. We have nucleus here. So this example of a bipolar neuron. So this is dendron and this one is axon. Okay. Example, very easy example, that is rod and cone cells of retina. So rod and cone cells of retina are basically bipolar neurons. And third is multipolar neuron. Most of neurons are multipolar. Most of the neuron are multipolar. Okay. So we have unipolar, we have bipolar, and we have multipolar neurons. And we have two more types of neurons. One is apolar, and one is pseudo unipolar. These are rare ones. These are the common and these are rare. Apolar neuron, they have no any process. They will appear like this. No any process. and they have pseudo unipolar. Basically, in case of pseudo unipolar, the axon is branched and it seems like there are two processes, but basically it is a one process. So this is an example of pseudo unipolar is dorsal root ganglion. We will talk about this when we will study spinal cord. Dorsal root ganglion. So this is the pseudo unipolar. Apolar, these are present in lower organisms. Lower organisms. And one is the another example is lacrimal. I think spelling is right. Lacrimal. Lacrimal gland is in our sorry amacrine. Okay. So this was all about the classification of neuron. From in next video we will learn about nerve impulse formation and conduction. So thank you and see you in the next video.